All right, time for the second part of this episode on my paper, Evolution of Multicellularity, Cheating Done Right. I'm sorry that it took quite a while to get this episode out. I had a summer break and was focused on work projects as well as travel. Now I'm back with these podcast episodes. And yeah, let's dive right into the paper. Let me share the screen. Here we go. So we stopped thinking about the theoretical aspects of this paper. How can we think about multicellularity evolving? How we something like a new individual merging on a higher level, constituted of individuals at a lower level? Um, and in fact, Rainey Mulroney, together with his collaborators, came up with an ingenious idea, originally from Rainey and Kerr. That's where the figure is from. Unfortunately, Springer did not accurately put the references out there, despite me requesting that in a proving stage. So there is a correction out there, but here you can see in the footnote 2010, page 875. And they proposed in this paper a new idea for the evolution of multicellularity, where you can look at these mats forming from individual cells, and in a way, they get destroyed. The individual, the SMAT, if you think about it as a new individual, inevitably ends, and there's no new life emerging from that, no new life cycle of mats replicating themselves. But you could think of a cheating cell that stops producing this glue, which leads to the formation of cells, separating itself from the mat and perhaps starting a new life cycle. And that's an idea in experimental evolution we shall look at now. So, evidence from experimental evolution. Rather than finding ways for optimal cheetah suppression leading to multicellularity, Hammerschmidt et al. 2014 test the hypothesis of Rainey and Kerr 2010 that cheetah suppression may in fact hinder the evolution of multicellularity, with cheats being necessary for the generation of a propagule. This enables a form of group reproduction which simultaneously solves the problem of ensuring the right amount of cooperation, providing both a mechanism of group reproduction and a mechanism to minimize the adverse effects of cheats. As illustrated by the debates on group selection, um, cheats have the potential to undermine the selection at higher levels, reaping the benefit of belonging to a collective without contributing themselves. This led to multiple researchers, in particular Michaud, in a couple of papers as well as a book, to argue for the necessity of cheetah suppression for multicellular organisms to emerge. Contrary to what common sense might suggest, cheetah suppression is not the only mechanism able to minimize the adverse effects of cheats for the group, however. As highlighted by Rainey and Kerr 2010, cheats may be necessary for the generation of a propagule and the enabling of group reduction. In order to provide evidence for this hypothesis, Hammerschmidt et al. Conducted the following experiment. See figure 2 up here. They propagated their bacterium Pseudomonas florensis in a spatially structured, undisturbed microcosms. That is, beakers filled with a nutritious broth, able to support the propagation depicted above. The original genotype with which the experiment starts has a smooth genotype expression with individual cells freely floating within the broth. Due to short life cycles of the individual cells, because they reproduce every hour and a high mutation rate, some of them quickly mutated from the ancestral smooth genotype, which I shall abbreviate as SM, to wrinkly spreaders, WS, following their abbreviations. So each labeled by the distinct appearance of their respective colony morphologies. Here's another nice figure from their paper. Again, Springer did not accurately put, unfortunately, the reference here. You can see it down here. Nevertheless, these wrinkly spreader mutants overproduced exaltated cellulose. That is a costly cell-cell glue, preventing their daughter cells also producing the cell glue from detaching themselves after cell division. Normally, such groups of cells have lower fitness, not only because the production of such a polymer is expensive in terms of fitness, but also because these associations take up space close to each other and thereby become competitors for resources. In the setting depicted above, figure 3, however, the phenotype here blue spreads because these cell glue associations are able to reap the benefits of access. Let me make this actually larger here for the viewers. Of oxygen staying at the interface between the air and the liquid growth medium, something the ancestral type was not able to achieve. As Rainey et al. 2017 points out, these mats are not buoyant and hence require a physical structure to attach themselves and reap the benefits of this ecological niche, further illustrating the importance of the environment and evolution of multicellularity. It is here the beaker. The mat gets bigger and bigger, taking over the whole surface, a niche previously unexplored. But selection on the individual cell level will make mutant cheats eventually prosper, leading to cheating cells, here green, reaping the benefits of cooperation without contributing to the glue necessary for the mat to survive. This spells doom for the Winkley's brother mats, as the public good, that is access to oxygen, cannot be sustained. While these mutants can stay at the top of the mat and reproduce, the mat gets heavier, while simultaneously lowering the integrity of the mat itself. When it collapses, all Winkley's brother cells in the mat are driven to extinction due to their lower fitness. This is a classical tragedy of the commons. This conflict between cooperating Winkley's brother cells and cheats, they argue, quote, could fuel evolution, 2014, page 75, by giving selection something to act on. As the Winkley's brother mats lack means of collective reproduction, they are similar to Zoma, an evolutionary dead end. While cheats do not spell doom for the mat, they might also be its savior. Figures, I shall briefly talk about them here. So, by becoming a germline of sorts, leading to a primitive life cycle, switching between Winkley's brother mats and mutant propagules. This may then enable mats to participate marginally as a unit of selection. As Godfrey Smith explained, a life cycle in a single cell propagule will be able to drive Darwinian mechanisms, which is what this experiment tries to capture. Let me now turn to a detailed analysis of the experiment responsible for the creation of this paper, see figure 4. Initially, Hammersmith et al. Only tested one regime consisting 120 of these spatially undisturbed microcosms. Every microcosm starts with a Winkley's brother genotype, here yellow. In phase 1, seen as our illustration above, each of the 120 microcosms is left alone for 6 days, after which the cells are harvested and screened for SM colonies. If no Winkley's brother mat is formed or no SM are found, the line ends, that it goes extinct. If no Winkley's brother mat is present, the multicellular body has not remained intact. While failure to produce SM cells, here red, stands for failure to produce propagules. So, in phase 2, which only lasted for 3 days, the microcosms continued solely with the newly formed SM cells. If they failed to transition back to Winkley's brother cells, within this time period, the line was also taken to be extinct. By analogy, imagine the failure of a propagule to develop further. After 6 generations, all lines were extinct. However, Hammersmith et al. Argue that even this persistence is sufficiently surprising and could bring these lineages under developmental control. They suggest that if successful lineages were allowed to take over the microcosms left by those lineages going extinct, it could allow, quote, the possibility for life cycle enhancing mutations, which are beneficial over the longer time scale of the life cycle, able to outrun life cycle disrupting mutations, page 76. Let's see figure 5. In response to this theoretical possibility, Hammersmith et al. Devised 2 regimes depicted above, consisting of one in which cheats were embraced and one in which they were purged, each of which consisting again of 120 spatially structured microcosms, however now divided into 15 populations of 8 lines. Both regimes went through a single cell bottleneck, one SM cell and one Winkley's brother cell, respectively. For the cheat-embracing regime, nothing changes from the initial experiment above, except for the fact that the microcosms left by extinct lineages are taken over by successful ones within the same population. Contrary to what one might expect, the cheat-embracing regime led to a life cycle-enhancing mutations, outweighing the effects of life cycle-disrupting mutations, while the cheat-purging regime did not show such an evolutionary response, as they do not have a proto-germline for Darwinian mechanisms to act upon. What may seem abstruse at first is perhaps a major step towards explaining the transition towards multicellularity. Rather than cheat suppression, cheat embracing simultaneously solves the problem of group reproduction and minimization of adverse effects of cheats. While cheats are detrimental to the fitness of the mad organism, without them there would be no group reproduction at all. As cheats are needed, the mad organism has to bring cheats under developmental control in order to move from multi-level selection 1 to multi-level selection 2, as discussed before. The Hammerschmidt et al. Experiment is then a classic example for survival reproduction trade-offs at the level of mats. While cheats lead to an earlier death of the mad organism, seeing them as germ cells makes it obvious that more cheats allow for a higher fecundity, that is, higher number of propagules. However, sometimes higher fecundity may be more valuable than higher survival, which is why the focus in the literature on mere cooperation among constituent cells has led to Rainey's new hypothesis. Given the right ecological conditions, cheats can justifiably be treated as both the doom and savior of the multicellular organism. The results suggest that witness decoupling is taking place in the cheat embracing regime, just as Rainey's theoretical work on the major transitions predicted. While the collective fitness of the MAD and the cheat purging regime is simply the average fitness of its constituent cells, that is multilevel selection 1, the fitness of the collective in the cheat embracing regime is taking its first marginal steps towards multilevel selection 2. Kasha 2006 or Scott v. Smith 2009 might put it, the MADs take the first steps to becoming Darwinian populations themselves. Interesting work on the relation between group formation by adhesion and evolution of group reproduction has been conducted using game theoretic models. Garcia et al. 2015 argued that cellular adhesion is a key ingredient to the evolution of multicellularity, as exemplified by the propensity of glue production by Winkley's predator cells. Furthermore, Tanita et al. 2010 have highlighted the importance of staying together, with daughter cells staying attached, thereby enabling further integration and the germline coming under developmental control. In addition, Pishugin et al. 2017 show such a multicellular strategy of fragmentation via single-cell propagules may often be superior to individual dispersion, making Rainey's hypothesis of cheats generating new life cycles all the more credible. In the cheat-embracing regime, the fitness of the mad lineages increases, while the fitness of the individual cells is actually decreased. The longer life cycle of the mad organism stands to conquer the selection among individual cells, making them work for the benefit of the group. Though the group reproduction via switching cheats is only a marginal case for reproduction, it gives evolution perhaps something to act on, potentially bringing the propagule under developmental control. After all, a small number of mutations in the original SM genotype led to the emergence of group properties in a proto-life cycle. Due to the artificial nature of the laboratory setting, some may doubt the validity of their results. To combat such claims and make the experiment more realistic, Rainey et al. 2017 suggest the example of a pond with randomly located reeds as both a thought experiment and a potential field experiment. As mad-forming cells are non-buoyant, they require something to attach themselves, otherwise not being able to reap the benefits of access to oxygen. The reeds provide one such environmental option at which the mads can form cooperative organisms, group organisms with higher fitness than individual ancestral types, hence what he calls ecological scaffolding. Mads here, just like in the experimental setting, inevitably fall to their doom. However, if a new germline by detaching cheats can be created, then a new life cycle at the same reed or another can begin, even allowing for the spread of these mad organisms. One important further environmental condition is the sufficient distance between reeds to prevent overlapping growth of mads and allow for variation, and hence selection between mad organisms. The two timescales between cell and mad reproduction generate two levels of selection, even though the Darwinian properties at the mad level are in the beginning only exogenously provided. That, however, is exactly what is needed in order to take the first step towards the evolution of a new Darwinian individual, with these properties becoming endogenized through the evolution of a developmentally determined life cycle. Quote, page 105. generating eco-evolutionary feedback loops. The importance of ecology has here not only the abiotic dimension, that is interaction among organisms between Winkley spreader and SM cell types, the smooth types, but also an abiotic dimension, interaction with the environment, which makes the modeling of these processes much more difficult. As Rainey et al. 2017 put it, quote, ecology is everything. The structure of the environment permits realization of Darwinian properties at a collective level, even in the absence of these properties being endogenously determined. Quote end, page 104 to 105. The Hammerschmidt et al. Experiment does exactly that, suggests possibilities for Darwinian properties to become endogenized. One such way was elegantly illustrated in the cheat embracing regime. Cheats evolved a simple genetic switch to secure the switching from somametes to germline propagules, marking the beginning of a new, though unstable, life cycle and the emergence of a proto-multicellular organism. The idea here is simple. Most multicellular organisms reproduce via single-cell propagules, something the origin of which is to be explained, and Rainey's hypothesis has the potential of doing so. Citing a study of eusociality, Novak et al., Tanita 2017 draws an analogy between Rainey's proposal and the eusocial colonies of ants. Both the mad organism and the colony have a differential lifespan for both the group organism and the individual members. Except for the queen in these ant colonies, however, all the individuals in the colony are sterile, making them slaves to the group, or as the name suggests, the queen. The newborn queens, Tanita points out, can just as much be interpreted as a cheat, attaching themselves once they have matured in order to mate, forming a new group organism. Naturally, some will respond that the newborn queens are in fact not selfish, but rather one integral part of the whole organism responsible for reproduction. Just as the detaching cheats have to switch back to glue reproduction in order to form a new mat, the queens need to form a new colony. It may not be reasonable at this point to treat these propagules as cheats, as they have an important role in the life cycle of the group organism. But there is an inherent ambiguity between the shorter life cycle of individuals and the longer life cycle of the group. However, this realization only came to be by questioning the very notions of cheats being always bad simply in virtue of their evaluative name. Traditional game theoretic analyses, excluding such roles for cheats, must necessarily cloud the generation of new hypotheses, granting them such an important role in the evolution of multicellularity. Tanita 2017 argues that it's precisely because the ecology and development of eusocial ant colonies are much better understood that we understand the transition to a new Darwinian individual. She further suggests that we stop using anthropomorphizing terms such as, quote, cooperation, cheating, and selfishness, biasing our study of eco-evolutionary processes, quote end, page 22. Now, though I deem it unlikely that these traditional terms can be cast off overnight, if ever, biologists need to be aware of the negative impact on the generation of new hypotheses. Let me now summarize the discussion and analyze the impact of Rainey's work for both conceptual issues and future work in experimental biology. Further, directions and conclusion. The primary aim of this paper was to illustrate that when it comes to the evolution of multicellular organisms and the emergence of new Darwinian individuals on a higher level, too much focus has been spent on cooperation alone. To understand the evolution of multicellularity, a conceptual shift in perspective is required. Cheats are no longer just a problem that needs to be solved for multicellularity to evolve, but rather two, as argued in this paper, the very key for the evolution of multicellularity. As Rainey noted, a mechanism of group reproduction and a mechanism to minimize the adverse effects of cheats are necessary conditions that might, together with cooperation, be sufficient for the emergence of a new Darwinian individuals. As I showed, this neatly matches onto the 3D space for Darwinian individuality Godfrey Smith proposed. A bottleneck during which a propagule marks the beginning of a new life cycle, a germline being specialized for reproduction of the collective and overall integration of the individuals, forming a new individual rather than just a group. All of these come in degrees, and the Hammerschmidt et al. Paper beautifully shows how the first steps can be taken towards Darwinian individuality. By seeing cheats as the first single-cell propagules marking the beginning of a new life cycle through a bottleneck, the problem of ensuring group reproduction and minimization of adverse cheats can be solved simultaneously. This introduces the germ-soma distinction and makes further refinements possible. Before such a probable can come under developmental control, a random mutation is necessary, allowing for a switching mechanism from a SM to a WS phenotype. The cheat purging regime did not manage to decouple MAD fitness from the fitness of the cells, while contrary to intuition, the cheat embracing regime captures the very first steps towards Darwinian individuality by showcasing that MAD fitness increases independently from the decreasing cell fitness. The cells start to work for the organism. Their carefully conducted experiment corroborates much of Rainey's theoretical work over the last decade. Their multi-level selection approach provides a strong evidence that cheats and conflict, contrary to intuition, outdrives the evolution of multicellularity, as opposed to cooperation. A sign that intentional notions such as cheats and altruism can cloud the generation of a fruitful hypothesis. It is unlikely that these notions will be replaced, perhaps due to an inherent desire to explain apparently purposeful behavior in biology or the importance of game theory into biology from economics, import, where intentional goals, of course, clearly play a role. See Rosenberg 2009. However, more emphasis should be put on the fact that cooperation is a technical term intended to be value-free, merely referring to phenotypes that increase the fitness of others, though associated with a cost to oneself. The importance of the environment here cannot be understated, for it calls into question the very labels of cheats and cooperators and highlights the importance of eco-evolutionary feedback. Because of these more than just curious results, Tanita 2017 even calls for revision of the orthodox cooperation framework, leading to misleading interpretations when the environment is omitted from the analysis's hand. Rainey's work may then provide a strong case for the extended evolutionary synthesis, highlighting the importance of the environment and the evolution of development. By largely omitting developmental and environmental factors playing a role in the evolution of multicellularity, cooperation and the suppression of cheats has been deemed to be more important than it actually is. However, as Libby and Rainey 2013 point out, the ecological conditions of multicellular organisms having evolved millions of years ago are, quote, essentially unknowable, page 1. Therefore, one should not be too quick in dismissing previous and extensive work in evolutionary game theory on the emergence and persistence of cooperation. The best we might be able to arrive at are robust, how possibly explanations of how multicellularity could have arisen. Furthermore, through further advances in experimental and synthetic biology, we may even be able to have multicellular organisms emerge in the lab. The analysis of environmental factors will help to narrow down the set of how possibly explanations and increase our confidence that proposed mechanisms such as the cheats as propagules hypothesis by Rainey may have been at work millions of years ago. Further work may focus on the impact of anthropomorphizing language on research. More attention should also be brought onto the inherent differences between eukaryotes and prokaryotes, as the most interesting cases of evolutionary transitions towards multicellularity tend to involve eukaryotes, even though the Hammerschmidt experiment provided evidence that even the prokaryote Pseudomonas florensis can take the first steps towards Darwinian individuality. Additional marginal cases for Darwinian individuality in the case of prokaryotes worthy of studying are social bacteria such as Coccus xanthus that form fruiting bodies with reproductive division of labor, see Crespi 2001, and filaments of cyanobacteria with cellular division of labor, see Rossetti et al. 2010. Furthermore, one could shed light on the developmental process involving proto-germlines coming under developmental control. Recent literature on size specialization feedback loops may provide further insight into the division of labor. See Borg 2011 and Birch 2017, or as Godfrey Smith 2009 calls it, integration, something that's still lacking in our account in the Hammerschmidt et al. Experiment, only marking the very beginning of a major transition in individuality. Also, as Rainey and Kerr note at the end of the 2010 paper, Heinegger 2002 has done interesting work on the aging mechanism, being a, quote, deprivation syndrome driven by the tension between the soma and germline, quote end, page 879. Once it is established that cheats serve as the origin of the germ-soma distinction, many research questions will be illuminated. At the very least, this opens up a new theoretical option that has largely been unexplored. Let me conclude this paper with a quote from the last page of Semio Kasha's Evolution and Levels of Selection. Quote, The study of evolutionary transitions is still in its infancy, with much empirical work remaining to be done. So it is difficult to say whether the foregoing analysis will prove satisfactory in all respects. But whatever future developments in the field look like, it is likely that multi-level selection will remain crucial for theorizing about evolutionary transitions. Quote end. 2006, page 240. And in this, Okasha's philosophical work on multi-level selection proved to be quite successful in generating a progressive research program. The experimental work of Hammerschmidt, Rose, Kay, and Rainey on the questions of how evolution transitions from singular cells to multicellular organisms is a prime example for the strength of the multi-level selection approach. In a mere decade, it seems, the study of evolutionary transitions underwent significant progress, offering a more positive view for the role of cheats and a recognition of the importance of ecology. Much work remains to be done, however, with prospects for the work of biologists and philosophers of biology blending together appearing more than promising. So, this was the paper. I hope you've enjoyed it. I certainly did. Unfortunate that even a few years ago, journals were already trying to save money and didn't put as much effort into the proving process. And then you have annoying errors appearing in papers. And I think that's becoming, unfortunately, more common now. Nevertheless, this was one of my very first papers. Started to write this during my undergrad, and I believe I did major revisions, minor revisions during my time as a master's student in Bristol. So one of my first publications in the philosophy of biology. And it was very interesting to me as, of course, coming from a background in philosophy and economics, to see how I could move more into the biology room where game theory, cooperation, these conflicts that use economic language can help us to understand biological phenomena. And I continue to do this in my recent work, where we can think about the evolution of consciousness related to this evolution of agency, of choice, of real utility functions that help organisms to make fitness-enhancing choices. But yeah, I hope you've enjoyed this episode, and I have another episode coming up soon with Peter Singer. See you until then.